Okay. Brilliant. Okay, so welcome those who've already joined. My name's Ellie Hay. I'm one of the clinical genetics registrars based at Great Ormond Street. And I have my colleague Shireen Tadros, who's also with us today, to talk to you about genetic testing options particularly, and a bit about the challenges of these, and also a quiz. We'd like to keep it as engaging as we can. So I'd be really grateful to you all if you could join in a couple of ways. So we have a Slido poll and some quiz a bit later on, which you can access either by going straight to the website and just popping in this number, the 41940, or hopefully the QR code would also work if you want to scan that. And then there are other places during the talk where it's probably easier just to put things into the chat. So you should all have the chat function on your computer, and then either Shireen or I can have a look at the answers that we're getting in there. And if you have any questions or anything you want to ask as we go along, then again, by all means do that. I think from a sort of housekeeping point of view, it's best if you can keep your videos and mics off and just enter things into the chat, particularly if we've got sort of over 20 or so people. My Wi-Fi is a little bit slow, so if things are not moving on for you, or you find that you can't hear me very well, again, do please put it in the chat and I'll see whether there's anything I can do. So we'll get started. I know many of you might have joined in for the lecture that Lara gave on Monday. So she was focusing mostly on dysmorphology and the approach to children with a potential genetic disorder. Today we're going to focus more on the different technologies which are available. And obviously I've only got an hour today. This is usually a lecture that I give over sort of 2 or 3 hours. So some of it will be a little bit brief, but where I can, I've added some links for you to have a look at. And then by looking at this, hopefully we'll be able to give you a better idea of what options are available for different types of conditions, and also appreciate some of the challenges that we have. And I'm not sure if any of you are aware, but there's going to be a new sort of national test directory, which is relevant for those in England. And I'll touch upon this briefly at the end. So we're having a look at the RCPCH curriculum. There certainly are various aspects sort of spreaded throughout, really, which is focusing on recognising and initiating investigations for a whole variety of different conditions, including dysmorphology, common conditions such as learning difficulties, and microcephaly. So I think it is important to understand these different types of testing that are available. But what would be quite handy for me is to have a bit of an understanding about what you know already. So as a sort of trial, we'll see whether we can have a first poll for Slido. So if you bear with me a second, let's see if we can make this work. Oh, perfect. Excellent. I think there are about 25 of you or so, so let's just give it a little bit longer and see whether anyone else is happy to contribute. It's all anonymous, so we can't see you, but it does really help us to get a bit of an understanding. This is not something I'd necessarily have thought that you would have had a lot of information about. It's not something that's taught on an undergraduate level, and in fact, even while I was in paediatrics, I don't think I had a very broad knowledge of the type of testing that was available. And of course, it's changing all the time. Let's see if we can get to 20. A couple more votes.
We won't know who you are.
Yeah, it's all anonymous. No, that might be all. Okay, so we've got certainly over half of you here. So I think, oh, a couple more. What I'm sensing is that, yes, you've got a bit of an understanding. So the majority of you going for not knowing what is available or knowing when to request an array, which is probably the most common genetic test that you're likely to come across in paediatrics. Brilliant, thank you for that. So let's see if we can move back. Oh, there was a bit of a lag. Okay, so this might be useful just if you can put some information in the chat. So why do we do genetic testing? So we have a real range of different families who are referred to us, and not all for the same reason. So what sort of things might they be referred to genetics for?
We've got one answer so far, prognosis.
Yeah. So thinking about working out what's family planning. Yeah. Counselling discussion. Perfect. Yep, those are the kind of things that we're thinking about. So we can often break these down into sort of diagnostic, carrier, pre-symptomatic and prenatal testing. We'll talk a little about those now. So I think many people think about it as being a diagnostic service, and that's certainly part of it. So when children present with perhaps an unusual combination of features and we think it might have a genetic cause, then we're performing a test to see whether we can confirm our clinical presentation. I'm sure these are sort of common conditions that you might come across in the paediatric world. And then from a carrier perspective, we're thinking about conditions which aren't necessarily going to affect the person who has come along. So they might have a child or someone in the family who's known to have a particular condition, but the risk for them would be for their offspring. And here we're thinking about autosomal recessive conditions such as cystic fibrosis, or the X-linked conditions such as Duchenne muscular dystrophy. And then a sort of more recent addition to genetics, and I think we alluded to it earlier on sort of counselling, can be pre-symptomatic testing. So this is not for a condition which a patient is presenting with features now, but for somebody who has no signs of the disease but might develop it in the future. And sometimes this can be important if we can intervene in some way, either by providing surveillance, screening, or with management, or because it helps them in terms of future planning. So things like Huntington's disease, or for the cancer susceptibility genes. And we don't tend to test for these in childhood if they're not going to present until adulthood. And there's a whole lot of sort of ethical concepts around this. It's an important branch of genetic testing to be aware of. And then finally, and I'll come to this a little bit later, is about prenatal testing. So this usually happens in the circumstance where you have a family, you have a child already with a genetic disorder, and they're thinking about future pregnancies and their risk. So that could be something that is a chromosomal, so an imbalance, or because of a single gene disorder. And there are various ways in which the prenatal testing can be undertaken. So the PGD, or pre-implantation genetic diagnosis. So this is when we are performing an IVF type procedure and then removing a cell at a very early stage to determine whether or not that embryo is affected, and then only replacing unaffected embryos. And then the NIPD is more, well, this is a very new branch really, looking at non-invasive prenatal diagnosis. So taking a blood sample from the mother and being able to detect the free fetal DNA and determining whether or not the fetus is likely to carry this genetic change. And then probably what you're more used to, the prenatal invasive testing, so things like chorionic villus sampling and amniocentesis. So, but the real focus of today is thinking about genetic testing, and there are clearly a number of different ways that we can do this. And I just wonder, can anyone give me a couple of genetic tests that they've heard of or that they've used themselves? We think about it broadly being split into sort of cytogenetics and molecular genetics. Microarray. So cyto is really thinking about the level of the chromosome. So are there any structural anomalies, or looking at dosage, too much or too little? Yep, karyotype fish, brilliant. Those are all examples of cytogenetics. Do you know any molecular genetics? So looking at the nucleotide level. Yep, NGS panels, brilliant. Exome sequencing. Yep, perfect. I think you've got most of them there. And we've got really sort of what's available in the NHS, and then there really are quite a number of research studies. The Deciphering Developmental Disorders and the 100K have closed now, but we're still getting results through from these. And it's an important way that we learn about sort of the next generation of sequencing or genetic testing before they move into the NHS arena. So we'll start with karyotyping then. So this is sort of the traditional way that we used to look at genetic material down a microscope. It is still used in a number of circumstances now, very much less so than before. So this is how we used to, and if you can see my cursor, so when we look at the cell itself, we have the chromosomes that are all scattered about, and some very, very clever clinical scientists can put these into the right order. I've never been able to do it myself. And really, you can determine whether there's an excess or a deletion of any of the genetic material, and also look at the position. So anyone want to hazard a guess as what we use a karyotype for now, if it's not standard for everyone with a genetic disorder? I know we have a bit of a lag, so aneuploidy, yep, it's looking, yeah. So looking at difference in the number of chromosomes. So, I went on a bit fast. But one of the main things we look for are these balanced chromosomal translocations. So don't know whether you can appreciate from this diagram, but we've got 2 copies of the chromosome 15 here, this one of which looks shorter, and 2 copies of chromosome 6, one of which looks longer. So what we have is a translocation of genetic material from 15 onto 6. But this patient is balanced because they've got the right amount of genetic information, albeit in the wrong location. So this causes problems predominantly when they go on to have children. So depending on how these chromosomes separate, you can end up with having a child with a normal contingent of the chromosomes, being a balanced carrier like their parent, or having an imbalance either with an excess or deletion of that information on either of the chromosomes. And that can either result in early miscarriage or a child who has a number of congenital anomalies, usually structural abnormalities as well as development. Anyone want to tell me what's wrong with this karyotype?
Some helpful arrows there.
Yeah, that's your clue. What's wrong here? Anyone see? Not a Philadelphia chromosome, but that is something that we can sometimes see on here. Robertsonian, yep. So what's Robertsonian? Translocation, yeah, absolutely. So here, one of the copies of 21 is missing, and here the chromosome 14 that's a lot longer. So the 21 has joined onto the 14. So this is, yeah, a Robertsonian translocation, and these occur on the acrosomic chromosome. So these are ones where they don't have a short arm, and so 13, 14, 15, 21 and 22 can join together. So you end up with aneuploidy. So these got 45 chromosomes, not 46. But this, again, is somebody who should be well because they've got the right amount of genetic information, but just in the wrong place. So again, the problem comes when you have children. I think it's shown on this next one, because it's difficult for these chromosomes to separate properly. So what you can end up with is, again, having the normal chromosome contingent, a balanced carrier like their parent, or a difference in the amount of information that you have on either the 14 or the 21 in this case. So most will result in miscarriage, but the most common live-born abnormality would be trisomy 21 or Down syndrome. It's a translocation down, which is important because it has a different recurrence rate to those trisomy 21s, which is when you get a sporadic extra copy of 21. So moving sort of down from the whole chromosome level to a bit more targeted, we have what we call a FISH. So this is fluorescent in situ hybridization. And essentially what you're looking at is a very targeted region of a chromosome, and you can light that up with some bright colors and identify chromosome anomalies like insertions or deletions. We used to use it for microdeletions such as DeGeorge syndrome, but it's largely been superseded by other technologies such as an array. But what you can see here is the green marker is telling you that we're on chromosome 21, and the area that we're interested in, on 22, the area we're interested in is this red marker here, and you can see it's present on this copy of 22, but not on this one. So they're missing the DiGeorge critical region on chromosome 22. So that's how they make the diagnosis. And as I said, it's not used as much now, but still on the neonatal unit, you might find that it is utilized. And I think anyone who was here on Monday thinking about our dysmorphology teaching, can anyone tell me what is the diagnosis in this case? So this is the result, the FISH results from this baby. Sounds? Yeah. Perfect. So probably the most obvious sign that you can see in this child, is the upslanting palpable fissures. And here we've got the green probe, which is on chromosome 14, and there's 2 copies. And then the red probe is on chromosome 21. So you can see you've got 3. So this is a trisomy 21. So it's not from translocation. This is as a result of 3 copies of chromosome 21. Okay, so the microarray, some of you alluded to knowing about earlier. So again, this is cytotechnology, in that we're looking for a dosage problem, so too much or too little information. We can look at a much more higher level, so super high resolution karyotype in a way. And I won't go into too much detail here, but the process itself involves taking DNA from a control and from the patient and labeling it with a different color. So when you mix these together, they compete to hybridize on the plate that you have, And if you've got an equal amount from the control and the patient, then you'll get a yellow dot. If you've got too much of the green or the patient amount, then you'll see a duplication, for example, then it will appear more green. And if it's deleted, you'll see more of the control sample, so it'll be more red. So you get an image that looks a bit like this, which is clearly impossible to read by eye, so we employ computers to do this. And they give us a very nice readout. So all of this blue data here is where the patient and the control data are broadly in line. And then you can see here there's a loss of patient DNA around this area. So this is a deletion on this part of the chromosome. The arrays, I've already tell you how much information there is and whether it's too much or too little. It doesn't show you where it is, and it's much more high resolution than a karyotype, but we still can't see down to the very low levels. So it's a very, very useful test, but it does have some limitations. And does anyone want to tell me what arrays don't show? Can you give me one example of what they don't show? SNPs. So you can, well, a SNP array can. So a lot of the time we do, I haven't gone into it here, but array CGH. But at GOSH, actually, we use SNP array. So you can see some for that. Yeah, balanced translocations is really important. So it wouldn't tell you whether you had a balanced translocation because all the information is there, albeit in the wrong place. Triplet repeats, yeah, absolutely. I think most of them here. So as you mentioned, very small changes. We can't detect mutations, so we're not looking for a base pair change or a mutation in a single gene. Balanced translocations and imprinting, and I probably won't have time to go into that today, but these are disorders like Prader-Willi or Beckwith-Biedermann, where the genetic anomaly is due to an imbalance of the maternal and paternal expression of those genes, although you might see it on the SNP array, as Ed mentioned. So when you get your reports back, they're not always that easy to interpret. So the first two are probably the ones that you know are easiest to understand. So we either haven't found anything at all, or we found something that's previously been reported, is well established, and we know what it causes. But a lot of the time, what we find is that people have copy number variants. So these might be something which is known to be a familial variant, in which case the lab might ignore it and not even report it. Or it can be a neurosusceptibility change, and I'll come to that in a minute. Or it can be completely of unknown significance, so it's never been seen before, and therefore it's difficult to establish whether it might be contributing to what the patient has presented with or be completely irrelevant. So I don't know if you heard of neurosusceptibility loci, but these are areas within the array which we've sort of increasingly understood to confer an increased risk of neurodevelopmental disorders such as ADHD, developmental delay, autism spectrum, for example. There's a few that I've got shown here. And what's important about these is that they're really difficult to interpret and difficult to discuss with families because they have incomplete penetrance. The penetrance is when you have inherited a genetic change and you show features relating to that. So incomplete penetrance means that you could carry one of these changes but not have any features of it whatsoever, or have very variable features either between families or even within families. And there's a difference depending on which one you have. So some of them will carry very low risk, so a 10% risk. So you might have inherited this change, but actually 90% of people with it don't have any problems. So they might have a parent who has exactly the same genetic change, but they don't have any neurodevelopmental problems. And that can be hard for parents to understand. Whereas some of them have got much higher risk, but it can vary. So the obesity risk might be quite high in terms of penetrance, but the developmental disorder perhaps less so. So one place that's really, really helpful to look is this website, which is Unique Rare Chromosome. And they produce lots of leaflets for professionals and actually for the families, which give a lot more information about the types of changes that you get and examples from families who have children with these conditions. And they're important to know about from a prenatal perspective because it differs between units as to whether they're reported. So in our unit, we don't report these prenatally because most people have had an array because they've got a scan abnormality, so a heart problem, a kidney problem. And these neurosusceptibility loci are not generally associated with those. So they're not going to be the explanation for the changes that you see in scan. The difficulty is that if you then do a postnatal array, you might get a different result from the prenatal because they then will be reported. But it can be difficult for parents again, because you're really saying to them that we found something, but we don't know whether the child will have any issues related to that or what those issues might be or when they might have them. So they are important sometimes for somebody who's presenting, say, with a neurodevelopmental disease, and we can say, okay, we've done this array and this explains it. But much harder if you've done it in the context of a different situation, so a scan abnormality or an early neonatal finding. Variants of unknown significance, again, the lab takes into consideration various things. So is it very, very big? So very large deletion is likely to be problematic because you're going to take out lots of genes. Or if it's something very small, it might not be. We have sort of gene deserts where they're patches of our chromosomes which don't seem to have an awful lot of genetic information. You can get away with a lot more. Also, it depends whether it's duplication or deletion. So in general, you can cope with a larger duplication than you can with a loss. But we do know that having too much of information can be problematic. So Down syndrome is due to having lots of extra information on chromosome 21. And sometimes we can look at the genes within these changes and understand whether or not it's likely to be significant. But it's also something else that we can do. So this is just testing the child. So what else can be useful when we've got these children with variants of unknown significance on their array? What else can we do? What other tests can we do? Thinking more broadly than the child. Yes, brilliant. So we test the parents, and that can be helpful, but sometimes it's not. In general, if it's a new change in the child, it's more likely to be contributing to the phenotype. But not, if we find it in the parent, doesn't necessarily mean that it's not significant. So if they have a similar sort of phenotype, then absolutely, it could be something that's seen in both the child and the parent. But also with these reduced penetrance ones, it can be more difficult to establish whether it's causative or not. But it can often be useful to do the parent. Okay, so that was sort of a whistle stop through cytogenetics. And now I'm going to talk a little bit more really about sequencing. So here we're looking really at the nucleotide level, so specifically more at genes. And there are various different approaches that can be taken, and depends how wide you wish to cast the net, really. But if we're just looking at a single gene, or even a specific recurrent mutation, then we might use a technology called Sanger. So this was something that was established first in terms of our sequencing. It's really helpful when you're looking at a well-established condition where we know it's only caused by one gene, so something like cystic fibrosis or Duchenne. Whereas the next generation sequencing technologies are better for when it's more genetically heterogeneous, so something like arthrogryposis, which can be a neurological or a muscular or a generalized syndromic disorder, or the epileptic encephalopathies, which can have a numerous number of genes associated with. Or we can go very broad, so pretty much agnostic. So look at anything that might potentially be diagnostic, either with whole exome sequencing, so looking at all of the areas which code for proteins, which is about 2% of the genetic information, or whole genome sequencing, so the entire sequencing code, including the bits between the genes, so all of our introns. And again, I don't have any time to go into the details of technology, but the Sanger sequencing is what we've been utilizing since really the 70s. And the principles of this have really involved chain termination. So you can see with some of the diagrams here. So you have a different color for your C, T, G, and A, and you mix your normal and your colored nucleotides, and they're randomly incorporated into the chain. And then you can run your gels, your electrophoresis, and use computer technology to read off which one of these colored letters is being indicated at each level. And then you'll have a code very nicely presented to you this way. And there are various advantages of Sanger sequencing. It really has a very good detection rate, including looking at mosaicism. So if you have two different cell lines, one with a mutation and one which is normal. And it's reduced cost if you're only looking at one gene, but that cost is obviously going to rise in succession if you do many genes on top of that, and it can take a lot of time. We still use it for a number of confirmations. So if we have a genetic change that's been diagnosed through one of the next generation sequencing technologies, gold standard would be to confirm it with Sanger. But probably more commonly, we use it for family screening. So if we detect a mutation in the family, we can test other members for it. And of course, do prenatal testing if somebody has a CVS to look for that familial variant. And of course, there's a much reduced risk of secondary findings. So we're only looking at one gene, so we're not at risk of finding out information about other things which are irrelevant to the presentation. As I said, the disadvantages are that you're limited to one gene, and it's not very good for conditions which are genetic. That's when we need next generation sequencing technology. These are the big guns. So each human genome is a unique sequence of 3 billion letters. So sequencing an entire genome is very, very complex, but it's massively parallel. So it's like doing many, many different Sanger sequencing tests with multiple genes. So you break down the DNA in a similar fashion, and you sequence it into reads, which you can see here. And then you line it to your reference genome and see whether you can detect any changes. So here the reference genome says that at this position we should have a T, whereas this patient has got C the whole way down. So this is different. And the first genome took 13 years to sequence, and today we can do this in probably about a day. And then we can store it. So although there are problems with the amount that we can store, actually potentially we could then go back to and reanalyze it in the future. So again, I won't dwell on how we read the data. This is something you would have covered in your undergraduate teaching. But what you need to know is what the message is, so what it should say. Are there any changes? And then be able to interpret them. So when we think about this, having a sentence, for example, and we look at the different changes that you can have on this letter C here, if you have a deletion, what you end up is a single letter change which completely disrupts the message. That could be the loss of a protein completely from this allele. Or you can get a change which results in that amino acid changing to a stop codon. Then you either have a shortened protein, or again, you lose the whole of that allele's production of protein. Or you could have a change which changes the meaning of the sentence, but perhaps not the structure. And this is perhaps more difficult to determine whether it's pathogenic. This could be something like looking at the function of an enzyme. We could have a single letter change that alters it slightly, but it still makes sense. And those are really hard to determine whether they might be causing a subtle change within that patient's phenotype. In general, it's clearer to decide whether a variant is disease causing if it shortens a protein or results in its complete loss. Missense are much more troublesome. And with that in mind, it really makes a difference as to how you decide what level of genetic sequencing you're going to do. So if you want to look at everything, then you've got the whole genome approach. And then we can move down, as I mentioned before, to the whole exome. So looking at the region which has proteins in it, the genes, and that causes 85% of our known mutations. Or we're looking at genes that we know the function of, or we just look at certain panels. And if you want to capture everything, then the whole genome approach is the way forward. But the drawback to this is that you have many, many more variants than you need, and it's hard to pull apart the one that is causative. Whereas the more focused you get, obviously you lose some of the wider genetic information, but you're more focused probably on the area that's likely to hold that genetic change. So as I mentioned, it's much, much cheaper to perform genetic testing now. So we're able to do it much more frequently. And it really is sort of understanding what the challenges are. So yes, I think the media representation is that you do whole genome sequencing and you can very easily pluck out that disease-causing change. But it isn't that simple, because with all that data, how do you find that causative change amongst the vast amounts of normal population data that you have? So there are challenges which are both technical, interpretation-based, but also ethical. So what are we looking for? We're looking for that primary diagnosis. So what explains the features that the child has come with? But then are there secondary diagnoses? So these might not be related to the presentation, but they might be important. So what we call incidental findings or actionable findings. So you might find that they have a adult onset genetic susceptibility to cancer, for example. Now, do we discuss that with them? That wasn't what they came forward for. But this is where sort of the ethics can be much more challenging within genetics. And then there can be information that's relevant for the parents, but not for the child. So we might find that there's a de novo change, so a new onset change in the child, which is the cause of their problem. Both parents are a carrier for a cystic fibrosis gene change. So that's obviously got relevance for their next child. And then we're also moving towards sort of precision medicine. So we're looking for things of therapeutic value. So we might find that they have changes within the genetic code, which means that they're more likely to have a adverse reaction to certain medications. The challenges are in designing the pipeline. So what do we pull out of the information that's been generated, reading that sequence and matching it to the reference genome, interpreting those variants. So what do we do with the ones where we've never seen them before? What do we do with incidental findings? And what do we do with actionable findings? We do have some guidelines on how we can approach these. And these are sort of UK, US based. And it's to try and make sure that everyone does it in a standardized approach. So it's too detailed to go into it here, but we look at how frequently do we see this genetic change in the general population who are well? Has it been reported in the literature before associated with this condition? Is it something that's predicted to have an effect on the protein? That sort of thing. And then we mix all these together to make a score, which we can then say, is this benign or likely benign? So class 1 or 2. Is it disease causing? So class 4 or 5. Or, as the vast majority appear to be, are they uncertain? So we don't have evidence either way. And these are important for many reasons, but one of the most important, I think, is to do with prenatal testing. So just to make sure you're all awake, I thought we'd do another poll. So let me see if we can see this one. So if we are doing prenatal testing, so a CVS for a family where they've got a child with a genetic condition, that we have a variant identified, what class can we use it for? So can we use it for everything, or are there certain classes that need to be looked at? Okay, we've got a couple now.
So just a reminder, class 5 is pathogenic, class 4 is likely pathogenic, class 3 is uncertain, 2 is likely benign, and 1 is benign. Come on, be brave.
Oh, it's going up. There's definitely a lag on this. What should we wait for? 20? Should we get 20? All anonymous.
Get 20. I mean, yeah.
Almost there. Well, I mean, it looks like what we're getting is the vast majority have gone for class 4 or 5. So we're looking at likely pathogenic, antigenic, and that is correct. So that's what we're looking for. We need to be certain that what we're testing for is the cause of the existing child's problem, because if it isn't, then we're going to run into trouble. And this is actually very often comes up thinking about privately funded, and it's not, because it's an ethical thing. If we're looking and we haven't got enough evidence that the genetic change we found is likely to be causing those problems, then we don't want to end up in a situation where somebody's having a termination for a change that is not causative in that child. Brilliant. Okay, so let's go back. Super. As I said, yeah, correct interpretation is crucial. So you don't want to undercall, but also you don't want to overcall, because it has implications for the child, for family members, and for prenatal testing. So again, I won't dwell on this slide, but it's trying to put it all together. So with the teaching that we had on Monday, thinking about history, examination, investigations, family history, what information do we have that we could put together and then decide which test is the best? What's our differential? What are we thinking it might be? And it's important to think about that, particularly when you do whole genome sequencing, because if you've thought this is going to be a Noonan type diagnosis and you find a change in a Noonan gene, you're much more likely to think, yeah, this is real. And then selecting the appropriate test. And we've been through all of these already, but it's important to think of the ones where you're looking for a change in a gene and those where you're looking for an abnormality in the amount of genetic information. And then we need to interpret it and think about how we interpret the variant that we think is going to be the cause, and those that are incidental, so those who are found by chance. And a multidisciplinary team meeting is the best way to do this, because you can have your clinical scientist, your clinical geneticist, and your mainstream physician. And then you can determine whether you think it's benign, so we give a negative report, not found anything, or pathogenic, so we have a diagnostic report, yes, it's the cause. Or sometimes you do a research report if it's somewhere in the middle, so we think there might be evidence in the future, but we've not got enough now. If you not find anything, then is it something else? You know, is it not genetic? Is it something a little bit more unusual, or have we done the right test? So if it's an imprinting disorder or something to do with methylation, you need a methylation test. You won't find it on anything else. If it's something like Fragile X, do you need to do triplet repeat testing, which again is not something that we can standardly do via whole genome sequencing? Or is it in the mitochondria DNA? So something like Leber's hereditary optic neuropathy. And so I just wanted to briefly touch upon what's coming soon. So there's recently been very large scale reconfiguration of genetic laboratories and the testing regimes. And they've consolidated to really a much smaller group of genomic medicine centres, which you can see. And that's so that we could standardise testing. So for example, all the ophthalmology testing will now be performed at Great Ormond Street. So everyone within England with a structural eye disorder will have their testing in our unit. So the same genes are reviewed and we develop an expertise around that. So the test directory is hoping to be launched perhaps in September, October. And there's directories for both cancer and inherited disorders. And this shows really which tests are commissioned on the NHS, what platforms are routinely used, and who is eligible. And this is a good link, actually, to have a look at some of those directories now. There are a lot of resources about this, and I think it's probably one of those things where you learn it as you go along and as you try it for yourself. But the Health Education England website is really very good. There's some links down here in talking clinicians through how they might request these tests and who might be eligible and what sort of things you need to go through in order to decide that. And if any of you are in paediatric intensive care or neonatal unit at the moment, you might have come across the Paediatric Rapid Trio Exome service. So there's about 700 tests a year that are commissioned, so that's what, 60 a month in England for diagnostic trio sequencing. So the child and both of their parents, And this is because it's a real gene agnostic approach and it's got a higher diagnostic yield. So previous research has shown that we can have, with the right selection, perhaps a yield of 30 to 40%. Although you might have to do other testing, such as the microarray or methylation studies, if you're also thinking about other conditions that won't be picked up on sequencing. And it's for children who are particularly unwell, who have suggestive features of a monogenic disorder, so a single gene cause, where the diagnosis would change their management. So it might be that it changes the course of their treatment that's offered, or perhaps they move from an aggressive treatment to a palliative approach. And these can be requested by local teams via clinical genetics service locally, and they use what's called an R14 form, and I'll show you one in a minute. And the local team can consent the families for this. This is what the form looks like, and it's quite cumbersome, but you can do it alongside your local genetics unit. And there's more information down here on the clinical test directory. And in the future, there will be lots of different types of testing that you can access via the test directory. And many of these are the ones that really were covered in your curriculum that we looked at at the beginning. And things such as congenital malformation or morphic features and intellectual disability are probably the most important, and you probably can't read it on here, but there are things that might say, so whole genome sequencing and array, or for intellectual disability, you might also have triplet repeat testing, and you can deselect these if you don't think they're relevant to the patient. So I've got a bit of a quiz, which again would be useful if many of you could access, and you can choose whether or not you put your name in. I can announce the top 3 people who do the best if you like, but we can also keep it completely anonymous if you'd rather. So let me see if I can move over to the polls. Right, start quiz. So really, I'm asking what would be the best test that we could proceed with children who are presenting with the little summaries that I've got up here. And again, you don't have to put in your name, so we can keep this completely anonymous. It will just come up as a digit, so we have a vague idea of how many people have answered. So think about this child. It's a very common presentation you might see on the neonatal unit. So they've got lots of different problems. They might have had their renal pelvic dilatation picked up antenatally. They've got a heart problem. Since they've been born, they've got some unusual features. They might be a bit floppy. They have extra fingers. They've been very small. So there's probably more than one appropriate response to this test, but just have a little think about what we're seeing here. So it's complex, multiple systems, lots of different areas. What could be the problem? Often these are the children who will then later develop developmental disorders or intellectual disability. Maybe give a little bit longer. We've got 11. Come on, let's get 20. At least half of you. 15. I can't check as we go along, so I have to. I'll see if we can get 20 and then I'll move it on.
We're almost there. You can always put a pseudonym in you.
Oh yes, that's true. 17. Oh, I think that will do. So let's have a look. So most people have said do an array, 70% do an array, and you are correct. So yeah, this really is somewhere where the array is an excellent test, and actually it's probably something we do on absolutely every child presenting with these sort of features, even if we thought whole genome sequencing might also give us the answer. And it's because we can now turn the array around really in the matter of a few days in an urgent situation, and often it will give us our result without having to do any more complex testing. So that would be an excellent first line. Well done. Okay, so slightly different one here. So again, a 6-week-old, so a neonate just about, again with some dysmorphic features, but they've never left the neonatal unit. So they've got severe congenital heart disease. We don't know why. They look a bit unusual. We're thinking, could this be a genetic condition? And the surgeons are saying, well, what have they got? Do we need to do surgery? We're not sure whether they'll survive. Is it going to be worth it? What might we be looking for here? Okay, we've got 10. We keep going. So you can get 17 again. Or perhaps we're getting a bit braver. 16. Lost one. 17.
How did we manage that?
Oh, wow. Okay, keep going. 19. Can we get 20? Nice round number. Okay. So rapid trio exome, so an R14 form. Do we think that's correct? Yes, brilliant again. So this is really somebody who might be eligible for this sort of testing, and it's really important if you're on a unit because it's very sporadic. Some units are very good at referring these patients to us, others I think just haven't heard of it yet. It is a patient who, if it can change management immediately, it's a really good idea to see whether you can get a diagnosis through a broader testing, so more agnostic. Okay, so how about this one? It's perhaps a little bit mean. So it's a boy, and the boy aspect is important here. And he's got learning difficulties, he's autistic. And you're talking about the family history, and the mother said actually she had premature ovarian failure. And she's got a brother who's been in residential care, nobody's really sure why, has been there since a child. So we're thinking about a specific condition here, which all of you will have heard of. Okay, we're rising. We can get to 15 again, I'm aware of the time. Oh, brilliant. Okay, excellent. So what do we think? Most people, again, over 70% have opted for triplet repeat testing. Yep, and that's completely accurate. So this child sounds like they have a family history of Fragile X. So you wouldn't pick that up on array. Increasingly, we are picking up on whole genome sequencing, but it's not reliable enough yet because these triplet repeats are hard to sequence because it's difficult, because it's short read to know whereabouts, you know, how long that repeat is overall. So the best way to test for it is by doing specific testing through PCR or other options. So that's brilliant, well done. Okay. So another couple to go. So this is again a baby, so neonatal unit, admitted, and he's just got low tone. So he's not feeding, he's got undescended testes, he's not particularly dysmorphic, there's no family history of note. And actually, in honesty, you would do more than one test on this child at a time. But this is just trying to highlight some of the differences in testing which are important. So this one's not necessarily going to change management per se. They're kind of feeding and growing, but they're not quite right. I'm trying to determine if there's something underlying this presentation. This is a bit mean, really.
Very nearly.
It is very common. It's a really good one to know about, though, if you're on the neonatal unit, to be honest, because you might actually diagnose one yourself. So a bit of a mix, and I was expecting that actually, because we would probably do a barrage of tests on this baby. So we probably would do an array. We'd probably look for testing for SMA, maybe muscular dystrophy type conditions. But what I was aiming for was down here was the methylation studies. So children can present with Prader-Willi syndrome. So it's one of the imprinting disorders, and they don't often have a lot to necessarily show other than they're very hypertonic and they don't feed very well, and they can have these undescended testes. So I think it is important to do an array. It's probably important to do other testing such as the SMA, so the floppy baby panel as we call it. But the methylation studies is really one of the only ways you can diagnose these changes in children, unless it's as a result of a deletion, in which case you'll pick that up on the array anyway. Okay, brilliant. And then the last one I've got here is just thinking prenatally. So it might be that you see a couple who've had a baby who's been terminated with a family history, and now they're pregnant again, and they've got a diagnosis of arthrogryposis, so more of a symptom really. So child isn't moving, has lots of contractures, and it's only been detected at the 19-week scan. So again, can you think of what might be an appropriate test in this situation? So this is not a specific diagnosis. Arthrogryposis could be neurological, it could be muscular, it can be associated with some single gene disorders. What might we have a look at? Brilliant. Good. Okay, good numbers. Let's have a look. So selected panels, 60%. Absolutely right. So again, this is one of those cases where you want to be broader, so you can't just look at a single gene because there are so many different types and causes of arthrogryposis, but you perhaps don't want all of that data because it would be hard to interpret. So you can look specifically at a panel of genes which we know can cause this presentation at that time. Great. Oh, there we go. Sorry.
That's great.
It's going to automatically come up. So congratulations to Carolyn and Reese, joint first place. Brilliant. That's our hardest question, apparently. Okay, let's go back. Super. So that was really good, actually. And I think you've taken an awful lot of that information, which was very rapidly delivered, on what might be the best approach to testing in these children. So I was actually going to, while I've got a few of you online, just have a poll really to see what sort of teaching you might want in the future. So I've been liaising with the RCPCH because they're keen to involve more genomics teaching within the curriculum. But we were wondering what the best way to deliver this was. So if you're able to give me any thoughts, that would be really helpful. Let me just see if I can bring up the right test for that. Yeah. So I'm aware that there are quite a lot of virtual platforms at the moment, and that you get quite a lot of good teaching in London, though I think it's probably less consistent in other deaneries. Is a regular genetics teaching session something that you would appreciate having, and how often would you like it? So is it something you'd like little and often, or could we present more of a sort of conference style teaching, but sort of maybe half a day? I think ultimately it's something that we'd like to have recorded so that you can access it at different times, because clearly it's not going to be suitable for everyone if it's at lunchtime or in the evening. But I think if we can canvass opinion from a few of you, even if we can get 20, that would be really helpful for us when we're putting suggestions forward to the college. Okay, well, I'll leave that one open and see whether we get more later. Let's just go back. So, how long have we got? We've got about 10 minutes.
Ellie, sorry, we've just got a question about the first question in the quiz. Yeah. Whether we could go over that again.
The first question in the quiz. Yeah, so when we were talking about a neonate who's got multiple congenital anomalies and IUGR, is that right? That was the first. So, to be honest, I don't know whether Shireen, you agree with me here. I think for the most part, the microarray is the first line test that we do in almost every child. Yeah. And that's because it's an easy test that we can do. It's accurate, and we can get a result in a few days' time, which is really beneficial. And quite often we see that children with problems in multiple different areas is as a result of a dosage change. So it's because you have a deletion or duplication of a large number of genes. Those genes are encoding many different systems. So it might be ones that are related to the kidney, to the heart, to the brain. Universally, these sort of deletions tend to cause small children, or IUGR, tend to lead to developmental disorders and intellectual disability, even if we don't know what genes are involved. But then also have this variety of different problems. So generally as a first line test, it's the one to go for. We might also do that alongside, say, a rapid trio exome if it was likely to change the management or outcome for a child, say on the neonatal unit or paediatric intensive care. Does that answer that question okay? Is that Heather?
Yeah, let us know, Heather, if there was something specific that you wanted us. Okay.
Brilliant. So I think I've probably exhausted your quiz time. So we've got some more, but I think I'll probably leave it there unless you're keen to. And we can answer any other questions if you have them. I mean, it's recorded, so hopefully all of the links will be accessible for you to go back to in the future. But yeah, do let us know if there's anything else you would like to bring up from anything we've discussed today.
Thanks, Ellie. Just yeah, let us know in the chat if there's any questions that are arising, or if you do want to have a look at those last questions that Ellie's prepared.
Yeah, I don't know, will it, maybe I have to go through it slowly so it will come up on the screen. But I've got my email address down here as well. How should we interpret this privately? Yeah, that can be really, really difficult. And it's causing us a lot of problems. I don't know, Tareen, do you want to answer?
No, I was just say what the question is in case everyone hasn't seen it. So the question is, how should we interpret genetic tests privately done, especially antenatal ones? I mean, yeah, it is a very tricky one. I think we in the UK have a system of whereby our laboratories are regulated, And so we wouldn't automatically believe a test result if it's not from a regulated lab. Usually in the UK, even if something's been done privately, it will be in an accredited lab. But we very often have people coming with results, particularly from overseas, where we would need to sort of clarify that result ourselves. And sometimes that can be tricky because it might not have been a test that we would have undertaken in the first place. So obviously there's issues there around, use of resources, etc., where it might not have necessarily been an appropriate test in the first place. I don't know, Ellie, whether you want to add something there.
No, I think that's completely, yeah, that's sort of us, isn't it? It can be really difficult, and it can be very difficult for the parents. But we do tests sometimes, and particularly with ones, it gets very tricky when people have done the sort of over-the-counter tests and ones where they've had prenatal testing done privately, because some of the tests that they do from the prenatal basis certainly are ones that we don't think are accurate enough. So they often have a poor positive predictive value, and therefore we wouldn't have even have commented on them. But we also don't have any reason to repeat them because they might have done them for their own security, not because there's a scan abnormality, for example. And I think this is something that we are debating more and more in our departments as to what level of repeat testing should we be offering. And actually these people who are doing it privately should really have their counselling and follow-up in the same private setting, but we know from experience that that's not always the case.
Yeah, and certainly with direct-to-consumer testing, it's becoming more of an issue in terms of the amount of NHS resources that it takes. I think Ed might want you to go over those final questions if you had any.
Yeah, shall I do it slowly? Let's go back up. Hang on. The answers are the ones. I suppose we can record it, can't we? And then they can play it later on, as long as we haven't got anybody after us. So do you want to do some, Shireen, do you want to talk about this one? Oh, hang on. So this is a report that you've got, which has got both your karyotype and your array findings, and it gives you a little bit of information here. And what we're essentially saying is, get it, that this is an unbalanced translocation, see between chromosome 9 at this breakpoint and chromosome 17. And you've got an extra bit on chromosome 17, no loss or gain of chromosome 9. And the DN here stands for de novo, so having occurred for the first time in that parent. Neither parent has a balanced translocation, so you're not expecting this is a high recurrence rate for the next pregnancy. And in this one, we're looking at the significance of this molecular test. So this is a patient who's been tested for neurofibromatosis type 1 and spread 1, which is Legius syndrome. And because they've shortened it here, Cathio lays patches, and they've been found to have a heterozygous, a change in one copy of the NF1 gene. And they've said that this is pathogenic, so disease causing. This confirms the diagnosis in the patient, and it means that we have the mutation available for testing other family members or to do a prenatal invasive test such as a CVS. This is, as we were saying before with prenatal, this is a class, I don't think you said it here, it's a class 4 or 5 because it's pathogenic. And therefore it is, you would be permitted to do testing. When we're looking at this one, this is a child who has, was an adult, I can't remember. So ABC has got changes in GJB2, so 2 changes, so on both copies. So they have connexin 26 related deafness, so the most common cause of deafness. And when you read through this, it tells you that we believe that both are pathogenic, so both disease causing. And so they've confirmed the diagnosis in this patient, but their partner, DEF, has also got connexin 26 related deafness. So they're both deaf and they've both got changes on both copies of this gene, GJB2. And the importance of that is because when they are going on to have their own children, they either pass on this copy of the gene or this copy. If both of them are deaf, it means that the likelihood of a child being affected is 100%. So they're always going to have a copy of that gene which has a gene change on it. They'll have 2 copies and therefore have connexin 26 related. This is going back to what we were talking before about karyotyping. So we've got a child who has Down syndrome and they have a DER, so a derivative chromosome. This is 2121. When we look at the mother, she's aneuploid, so she should have 46 chromosomes. She's got 45, and that's because she's got her 22s just stuck together. And again, she's got the right amount of genetic information in the wrong place, so it doesn't cause her any problems. But she can only either pass on the derivative chromosome with both copies, or the copy that has no chromosome 21. So this is a Robertsonian translocation, so the acrocentric chromosomes. And the recurrence risk for this lady, unfortunately, would be 100% because she monosomy 21, or rather, passing on no 21, would result in miscarriage. And then this is just a useful one, thinking more broadly. So this is a child who has a clinical diagnosis of cystic fibrosis, and they found 2 different mutations. So they've inherited mutation A from the mother, but mutation B hasn't come from the father. So in theory, this could be de novo, so a brand new mutation, CFTR gene. That's really, really rare. And actually the most common cause is that it's non-paternity. So this is not the, and that can cause ethical problems and challenges. But also it's really important when you're thinking about whether you're going to do a prenatal test in a following pregnancy, because actually the risk to this child is, or to the next child, sorry, is none, because this is not their father. And then this was talking about more of the neurosusceptibility loci. So can you offer prenatal testing? And that will only tell you whether the fetus has got the deletion or not, but won't predict the severity, and the penetrance is perhaps 10%. So then you get into, again, more ethical dilemmas, deciding whether or not this is something that you would offer a termination of pregnancy if there was a low risk of the fetus having. And then finally, we just had some implications of the cognitive variants. So if you have a dysmorphic child with delay who you find has got a deletion on chromosome 14 with involving this gene, which can cause early onset Alzheimer's if there's a mutation, so that's not a deletion, what would you do? It's obviously not going to affect the child and it's not the cause of their problems. So again, it's one that's open for discussion. So deletions might not have the same effect, so it might be completely irrelevant. And it'd be more worrying if it's been deleted from, inherited from a parent who has a family history of early onset Alzheimer's. Disclosure aspect is challenging and something we'd be doing in an MDT setting. And then I think this is the last question, thinking about Huntington's. So we have a boy who has been found to have a progressive neurological disorder. They've been tested through a 100,000 Genomes project, so one of the research projects I mentioned earlier with whole genome sequencing. And they found an expansion in the Huntington's gene, which would fit the phenotype, though is unusual as a juvenile presentation. So the parents are in their 30s and aren't aware of the history. So thinking about the explanation for this boy's result, well, it could be anticipation, so the sequence expanding in subsequent generations, or non-paternity. So the parents are asking about themselves, what would you do? So this has got obviously massive implications for the family and something that you'd want to seek genetic advice about if you got the result. Because there are set protocols that we follow with the pre-symptomatic testing, as I mentioned before, about deciding whether it's the right decision for you. We wouldn't ordinarily test children pre-symptomatically unless there's something that we can do about it. And currently there is no treatment or intervention that can slow down or prevent. So hopefully we've met the objectives for today. I know we've gone a little bit over, but as I mentioned, there are some really good links, particularly looking about the National Test Directory. And if you have any other questions or you're interested in genetics, then by all means do email me and I can do your best. Thank you all for your attention.